Our next speaker is Heran Darwin, professor of microbiology at the NYU Grossman School of Medicine. She received both her undergraduate degree and her PhD from UCLA. Mycobacterium tuberculosis, the germ that causes the disease tuberculosis, is an unusual pathogen. It only infects humans, and it's been a leading cause of death for millennia, currently killing more than 1 million people per year. Curative TB treatment takes months, and antibiotic resistance is a growing concern. So there's an urgent need to find new approaches to combat infection. Dr. Darwin studies the breakdown of proteins by specialized cellular machines within TB bacteria to understand how protein degradation is involved in infection and how it might be manipulated to develop new treatments. Elected to Class 4's section on microbial biology, Dr. Darwin's title is What Has Evolution Taught Us About Immunity to Infection? Dr. Darwin.
Okay, so first of all, thank you, Nancy, for the introduction. Thank you to the Academy for this amazing opportunity to just give you a very brief view of what has been going on in my lab for the last almost 21 years. And before anyone asks, I am not related to Charles. Okay, I need the clicker. Okay, so, all right, so first, just the brief, where was I born or however it goes. My scientific journey. So I was born in Santa Monica, California, to Korean immigrants seeking the American dream, like many other immigrants. And as the child of many immigrants, especially Asian immigrants, I was a pre-med at UCLA. And what happened when I was there was one of my colleagues who did become a physician said, we'll have a better chance of getting into medical school if you do a little research. So I went to a lab, the lab of Dr. Joan McEwen, who was working on baker's yeast. And I was immediately hooked, and I just ditched the idea of going to medical school, and I decided to apply to graduate school. And I stayed at UCLA, where I worked with Virginia Miller, who was working on Salmonella pathogenesis. So about halfway through my PhD, Virginia met the love of her life, Bill Goldman, who was a professor at Washington University in St. Louis. So she picked up the lab and moved it to St. Louis, and I followed her there. During which time, I had also met the love of my life, and he was a postdoc. He became a postdoc in the Miller Lab, and he got a job at NYU School of Medicine in 2001, so I followed him. So I had to find a postdoc, and initially I was going to do a postdoc at NYU working on cell death. But for reasons that we don't have to get into here, I ended up having to go to another lab, and I started to work with Carl Nathan at Weill Cornell Medical School to study tuberculosis. So after 3.5 years of doing my postdoc, I was very lucky to get a job at NYU School of Medicine as well in the Department of Microbiology in 2004. So this is a long journey, and I want to give extra credit to the people who were instrumental to bringing me here to this stage. So Virginia, I want to thank her for teaching me to be organized, rigorous, and careful. And she's also an amazing baker. Carl, for really teaching me to be bold and to also reach out to collaborators when you need it. I think collaboration is the best part of doing science. I really want to thank my former chairman, Claudio Basilico. This is very much him in situ with a glass of Prosecco and a cigarette. So Claudio, he took a chance on hiring me to the Department of Microbiology back in 2004. And in his department was the amazing Jan Vilcek, who is my friend and colleague, and he always got my back when I needed it most. And then most importantly, I have to thank my husband, Andrew Dirk Darwin, for always believing in me when I didn't always believe in myself. So thanks to the Jedi, and now for the science. Okay, so as you just heard, I work on Mycobacterium tuberculosis. I'll refer it to as TB, MTB, or TB bacteria. It is an airborne pathogen, and as you just heard, it is a human-exclusive pathogen that is transmitted only between people. You can infect small animals to study it in the laboratory. It is highly infectious. 10 bacilli are enough to infect a person, and as you just heard also, it is the leading cause of death in the world by a microbe. So 1 to 2 million people a year, and this has been happening probably for thousands of years. And one of the big problems with this disease is we do not have a reliable vaccine. Many of you in this room probably received a vaccine called BCG when you were babies. It does not work very well, and therefore in this country we don't even use it. And if you do get the disease, if you get infected, you have to take antibiotics for a very long time, 6 months minimum, during which time you cannot drink alcohol. So some people will actually say, I would rather not take the antibiotics. Seriously, I know things, okay? So we really need new treatments to think of better ways to treat this disease. Okay, so one of the reasons, so in order to find new treatments, we have to understand how is it that TB can somehow sit in our bodies for our entire lifetime until we have a weakened immune system. And we don't really fully understand how TB are controlled by the immune system, but we do know a couple of things. Okay, so what do we know? So in Carl Nathan's lab, Carl Nathan, now almost 30 years ago, discovered that macrophages, which are our first line of defense against infection, when they eat bacteria and they see a cytokine called interferon gamma, they will increase expression of many different genes, including one that encodes something called the inducible nitric oxide synthase, which makes a gas called nitric oxide. And nitric oxide can directly kill or inhibit bacterial growth. So we know about this. But despite our macrophages making this nitric oxide, the bacteria are not always sterilized by the macrophages, and that's why it lingers around in humans for decades. And the reason why, at least one thing we know, is that TB bacteria use a regulated protein degradation system to resist nitric oxide. And so as a postdoc, this is what I found was important for NO resistance, and then when I started my lab, we started to try to just understand the biochemistry of this system. So the system that my lab discovered and works on is called the PUP proteasome system, and it protects TB bacteria against nitric oxide death. So you have a protein that needs to be degraded, and a small disordered protein called PUP is attached by a ligase called PAF-A to make an isopeptide bond. This is a covalent leakage to a doomed protein. And then PUP is recognized by an ATPase attached to a protease called the proteasome. This looks very much like the ubiquitin proteasome system, except that that PUP and ubiquitin are biochemically very different. So this is probably why I'm here on this stage. Because PUP, or prokaryotic ubiquitin-like protein, was the first known protein-on-protein modification to be found in any prokaryote. And this is my first graduate student, Mike Pierce, who discovered this. And we know that dozens, if not hundreds of proteins are pupillated and degraded by bacterial proteasomes. So then this raised the question of which degraded protein or proteins are important for resistance to nitric oxide. And so an amazing postdoc, former postdoc in my lab, Maurice Simanovic, had found that it was the accumulation of a single protein called log that makes bacteria very sensitive to nitric oxide. So if you get rid of your proteasome, log accumulates, and log makes the bacteria very sensitive to nitric oxide. Okay, so what is log? Log stands for lonely guy, and I did not make that name up. It is an enzyme, and it was first described in plants, okay? And log mutations result in flowers with only one stamen and no pistil, right? So the male organ is the only thing there, no female organ, therefore it is a lonely guy. So the flowers look kind of janky like that. Okay, so lonely guy is actually conserved in a lot of bacterial species as well as in some other eukaryotic organisms. And what that does is it takes a molecule that looks like AMP, adenosine monophosphate, and it frees the base so that you have this adenine with this nitrogen 6 modification. And the final molecule is called a cytokinin. And what this does is it causes transcriptional changes in plants that help it develop. And plants are crazy because they have multiple copies of this gene, whereas bacteria just have one. And what Marie found is that cytokinins do signal transcriptional changes in bacteria, but it has nothing to do with nitric oxide. So we're just going to put that away for now. It is cool that TB does this. But what Marie figured out was she was looking at the literature and she found that cytokinins have to be enzymatically broken down back into adenine, as well as whatever comes off that N6, which is always an aldehyde. So proteasome defective strains accumulate lonely guy, they accumulate cytokinins, and they accumulate at least one aldehyde. Aldehydes are very unstable and reactive molecules, and so it's hard to detect these by mass spec, but we could at least see one of them. And aldehydes are often very reactive and toxic, right? So what we figured out was that basically the aldehydes were probably doing something to make the bacteria more sensitive to nitric oxide. So lonely guy needs to stay lonely. So in the absence of proteasome function, lonely guy accumulates, it makes cytokinins, it makes aldehydes, and then it makes damage. We don't know what all that damage is. And under normal growth conditions in the laboratory, the bacteria seem to tolerate this. But in the presence of nitric oxide, and now we know copper, the bacteria are dead. Okay, so meanwhile, at UC Berkeley, I did a sabbatical in 2015 at UC Berkeley with Russell Vance. And I met Sarah Stanley there, who is now one of my best friends. And at the time, she also studies tuberculosis, but she's looking a lot on the host side as well as the bacteria. And what she found was that macrophages that are infected with TB strongly increase aerobic glycolysis. Okay? And this requires interferon gamma, that really important immune cytokine. And so glycolysis makes several aldehydes, all right? And this is what kind of got us together to really think about this crazy conceptual leap, and that is maybe we make aldehydes to help control bacterial infections. And this is what we call the aldehyde hypothesis. Okay, so where did this idea come from? Well, I just spent the last 10 minutes telling you, 5 minutes telling you that in TB bacteria, if you lack a proteasome and you accumulate aldehydes, you become very sensitive to copper and nitric oxide. And I just told you Sarah found that immune cells that strongly increase glycolysis, which makes a lot of aldehydes, and including aldehydes that can kill bacteria like TB. Okay, but is there evidence in humans that this could be a thing? So I'm going to take a survey. How many people in this room, when they have a drink of alcohol, get all red and their eyes get all squinty and they feel really sick? Come on, there's got to be someone in here. I'm not, okay. Okay, there's definitely allergies to wine, but everybody in this room knows someone who drinks alcohol and they get very red and sick, and it's called the Asian flush, right, or the Asian glow. And this is a very common human genetic mutation called the ALDH2 star2 allele. It is in 500 million people in the world. It is probably the most common human genetic mutation on the planet. It is heterozygous dominant. And if you mutate this, if you carry this gene, you cannot metabolize a lot of aldehydes, including the aldehyde that comes from ethanol consumption. So you accumulate aldehydes, and it turns out about 10 years ago, a paper came out that showed that carriers of this mutation were less likely to have tuberculosis, okay? And in fact, we confirmed this with 2 other cohorts from Singapore and Vietnam a couple years ago. So, could disruption of the ALDH2 gene result in resistance to bacterial infections? And so, Russell Vance's lab so I'm going to start with this. This is a survival curve of mice. So, SP140 is a very important locus for resistance to not only TB, but other bacterial infections. So, these mice are incredibly sensitive to TB infections. So, these mice were made by Daisy G, a former student in the Vance lab. And what Sarah's lab did with her postdoc Sam Berry and student Scott Espich was they added the ALDH2 mutation onto this mouse background to see if it could protect it now from TB. And as a matter of fact, these mice lived longer because they had the ALDH2 mutation. And so, what I'm hoping to convince you is that aldehydes are a newly appreciated antibacterial effector. So the macrophages make nitric oxide, but they also induce aerobic glycolysis, which makes things like glyceraldehyde 3-phosphate, and methylglyoxyl, which is an aldehyde. What I didn't talk about is that we all also know that, well, most of us know that macrophages make reactive oxygen species, and these make incredibly toxic aldehydes that are lipid aldehydes called 4-hydroxynonenal and malonyl dialdehyde. And the enzyme that detoxifies those aldehydes is ALDH2. And what I don't have on this slide is we also have strong evidence that the aldehyde formaldehyde accumulates in activated macrophages. So we're really excited about where that's coming from and how the detoxification process occurs, and also how do the bacteria deal with that. And so I just want to end with this really important lesson that making connections between seemingly unrelated areas can be incredibly exciting. And so if it were not for discoveries in the ubiquitin proteasome system, plant biology, fundamental metabolism, and aldehyde chemistry, we could not have made any of these connections. And so I just want to thank the people in my lab. I don't think I have as many people as Flaminia has had. I've always had a small but mighty lab. We have very high specific activity, and I'm so grateful for all of them. And I'm really grateful for you for listening, and I'm happy to take questions.
And we do have time for several questions.
Don't be shy.
I'm Ellen Rothenberg from Caltech, and I'm from the opposite end of immunology, sort of molecular development. But I was very interested in this talk. I thought it was beautiful. And one question I had was the heterogeneity of the outcomes of the infections of macrophages by the MTB bacillus. And the sense from work of Joanne Flynn and others that when you look in, say, a primate model of tuberculosis, you see a lot of successfully contained nodules in the lungs, but a few that break out. So is there any link that you've found between what is actually going on in the regions of those nodules that are going to break out and the kinds of resistance mechanisms you're talking about? Because the bacteria are there in the successfully contained nodules, but they're not able to act.
Yeah, so I mean, the short answer is no, I don't know any of that. I think heterogeneity of infection, it's going to depend within one person, among a population, and we're only just starting to understand, well, what are the actual things that kill or control the bacteria? And the thing is even with the aldehydes, with nitric oxide, immunity, adaptive immunity is important. You need the cytokines, you need T cells, you need all that maintenance, and with age, those things start to wane, and that's when the bacteria wake up. But the bacteria, in my opinion, are constantly sampling and trying to understand, is it safe to come out, is it not safe to come out? But the heterogeneity with it, I mean, Joanne's going to have to answer that question, but thank you.
No, thank you.
Yeah, you're welcome.
Excellent.
I'm Luca Comai from UC Davis. Thank you for the shout out for plant biologists. I have a question about the persistence of this bacterium in the patient. Is this something that bacteria can do? I mean, you're talking about decades of survival of this bacterium in a latent state. What is known about it? I mean, what's the $1 million
question. Well, like I just said, I think the state of the immunity of the host is really important, right? So if you're healthy, the idea is your immune system is able to keep the fight going. I don't believe bacteria just fall asleep. I don't believe in dormancy in the sense that the bacteria are just, I think they're constantly sampling and trying to understand what's going on. I think dormancy is an active process in which the bacteria might shut down certain things. How it does that, I mean, that is the $1 million question to the field. I think because we don't know what all the when I say effectors, I mean things like nitric oxide, like aldehydes, like reactive oxygen species, like copper. All those things play into it, but we don't know the full scale, and we don't know how much each of those contributes to keeping bacteria suppressed. I don't know if that's satisfying for you.
My name is Keiko Tori from UT Austin, and thank you so much again, together with Luca, that shouting out for plant biology. I'm a very good friend of Junko Kyozuka, who discovered the log mutant.
Oh, great, cool.
It's scientist. Anyway, my question is, so you have this proteasome system. If you mutate, then log accumulate and make too much cytokinin, correct? I forgot. Did you check what's the function of the log knockout in TB? Does it have some sort of a fitness consequence in TB, or some infectious?
Yeah, no, it's a great question. So log is conserved in pathogenic mycobacteria. We cannot find a single animal-associated phenotype with either making too much log, no log. The gene that log actually is important for turning on. We can show, so mycobacteria are, they're not gram-negative or gram-positive, if you all know what that means. They have a very weird membrane that you use a special dye called acid-fast dye. And if log is there, it turns on a gene that makes the bacteria lose acid fasting. Now, acid-fast staining has been associated with pathogenesis. So I was excited. I'm like, this thing is going to be totally attenuated and we're going to, win all the big prizes. No, there was absolutely zero phenotype associated with it. So we know it's a signal transducing molecule. We just don't know what the downstream genes are important for, unfortunately. But yeah, no, it's a good question.
Thank you once again. Terrific.